Good morning, students. In this video, I explain development of endosperm and embryo after fertilization events contains post fertilization. In last video, I explained pre-fertilization, fertilization and post fertilization. Pre-fertilization, male and female gametes are formed. Fertilization, male and female gametes are fused. And last, post fertilization, development after fertilization contains post fertilization. In this process, development of endosperm and embryo. The development of endosperm. Endosperm developed occurs before the development of proper embryo. This is because the development of embryo requires heavy nutrition which is supplied by endosperm. So endosperm is sterile but nutritive tissues which provide the heavy nutrition of embryo. Then embryo development condition in properly occurs. After fertilization, the primary endosperm cell divides rapidly and forms triplide endosperm tissues. These tissues reserve heavy nutrients or large amount of reserve food. These conditions utilized by the cells of embryo for their development. In the most common types of endosperm developed in free nuclear endosperm, therefore developed cellular endosperm. Cellular endosperm, the upper covering of free nuclear endosperm. After endosperm development contains embryo development. Here we explain various stages of embryo development in dicot plant. You all know that what is dicot plants, which has two cotyledons called dicot plants. Here we show all stages of embryo development. After double fertilization, each ovules developed into a seed. Seed is the ripened ovules. It contains an embryo. When fertilization is complete in plant, this fertilization is called double fertilization. So, after endosperm development, embryo develops starting. Embryo develops from zygote. Zygote is the diploid cell which forms fertilization of male and female gametes. Embryo is developed at the micropylar end, which is zygote is situated. Micropylar end is the part of ovule. So this structure, this is the micropylar end. This part, zygote, is situated. The development of zygote into an embryo take place only after certain amount of endoplasm is formed. The earlier stages of embryo developed embryogeny are similar in both monocotyledons and dicotyledons. Embryogamy is the first development part of embryo. Both embryogamy are same or starting part of embryo development, those called embryogeny or embryogeny, is similar in monocotyledons and dicotyledons, but their seed are different after this process. The zygote gives rise to pre-embryo and globular, heart-shaped and mature embryo. These are the heart-shaped globular mature embryo. It is the last part of the embryo development. So here we started the development of embryo. The first stage, zygote. Zygote is situated in micropylar end of ovules. Zygote divides in two parts. First part called basal parts or basal cell, and next part or last part called terminal cell or terminal part. In the next step, basal cells divide. Transverse division form many cells. These basal cells divided in many cells 1, 2, 3. And arrange into row, this and this, and terminal cell divided in longitudinal and round shaped structure. It arranged all cells are round shaped structures. Arrangement of basal cells in linear shaped called suspensor cells. These cells are all cells are suspensor cells. Suspensor cells absorb maximum food because they are many numbers. It contains many types nutrients, so it absorbs the maximum food of endosperm and nutrients. The lowermost cells to row in different and differentiate into hypophyses. It gives the radical part of seeds and arranged two tiers of 4 each, 1, 2, 3, 4 and 1, 2, 3, 4. It total 8 in number. Upper tier cells in 4 in number, which support in suspensor cell, and lower tier gives rise the epicotyl, cotyledons and plumules. It make the larger part of the seed and it support the suspensor cells. This structure called pre-embryo because these are the half developed structure. These 4 cells support the suspensor cells and provide heavy nutrients with the help of endosperm, and last is made cotyledons. In cotyledons make two parts or two cotyledons are formed, but in monocot seeds only one cotyledons are formed. These structure called cotyledons. These part divided in two parts called cotyledons, and these part are the formed plumules. By this time the integuments of ovules become hard. All these part of the soft portion, and after this part seed develops a hard part, and this portion called seed and make seed coat. Then entire ovules is now called seed, and this after this process called seed. When the seed germinates, the cotyledons are used up to providing nourishment. The radicle gives rise root system. These are sometimes or after seed germination, these suspensor cells are reduced and mature embryo or develops, and cotyledons are well developed and it convert to seed. So seed is the ripened ovules. The ovary becomes, after seed germination, ovary becomes the fruit. The seed is internally contain maturing embryo. Then, in addition to embryo, each seed is covered with protective seed coat, which are derived from wall of ovule. The seed is dominant embryo saprophytic with the stored food and protective coat. Seeds are mainly two types. So here we complete the germination of seed from zygote. This process is called development of embryo or various stages of embryo. After maturing, the embryo form seed in the ovule. Seed are mainly two types: albuminous seed and non-albuminous seed. Albuminous seed called endospermic seed. In these seeds, our endosperm is consumed up slowly for nutritive purpose, and food storage system. And non-albuminous seeds are non-endospermic or X-albuminous seeds. In albuminous seeds are pea, groundnut, and non-albuminous seeds are mainly dicots plants. For example, gram, mustard, and some are monocots. For example, wheat, maize, barley, etc. Next is the fruits. Fruits are forms as the result of development of ovary walls and other flower parts. In simple words, a fruit is a ripened, mature ovary. The seed mature, they release the hormones which stimulates the walls of ovary to develop into fruit. After an ovule is fertilized, the ovary begins to expand. Fruits are distinct in many layers. First is the exocarp, exocarp of the outer layer of the fruit. Next is mesocarp, it is the middle layer of fruits, and endocarp is the inner layer of fruits. Fruits are mainly two types: true fruits and false fruits. And other type of fruits is the parthenocarpic fruits. So all over types of fruits are three: true fruits, false fruits, and parthenocarpic fruits. A false fruit is that which do not develops from the ovary or in which the significant part of the derived from floral part. For example, petals, sepals, or stamens, or other than ovary. For example, apple, cashew nut are the false fruits. And development of fruits without fertilization is called parthenocarpy fruits, such as banana are the most important fruit of the parthenocarpus process. These fruits are seedless fruits. Fruits are developed of the ovary wall and other floral parts. In simple words, fruits is a ripened or mature portion of the ovary. Fruits are mainly three types: true fruits, false fruits, and parthenocarpy fruits. True fruits formed developed from the ovary of a flower. You know that ovary is the female parts of the flower and lower part of the flower, and true fruits developed from single or ovary of the flower, and false fruits developed from the floral parts other than ovaries. For example, apple, strawberry, and cashew nut. And parthenocarpy fruits are developed without fertilization or without fertilization developed fruits called parthenocarpy fruits. Its fruits are seedless. For example, banana, pineapple, cucumber, grapes, orange. These fruits maturing the parthenocarpus process. So these fruits called parthenocarpy fruits. True fruits divided in three parts: simple fruits, aggregative fruits, and multiple fruits. Simple fruits developed from single ovary of a single flower. When a single flower presents single ovary, it develops simple type fruits. Simple type fruits are two types: dry fruits and fleshy fruits. Dry fruits upper part pericarp is dried in condition. You know that fruits divided in three parts: exocarp, mesocarp, and endocarp. Exocarp called epicarp or pericarp. This part of this type fruits is dried condition. So these fruits called dry fruits. For example, guava, orange. These is called dry fruits. Next is the fleshy fruits. Pericarp is fleshy, moist, or fibrous. For example, groundnut, mustard are the fleshy fruits. The next type fruits is the true fruit is aggregative fruits. These fruits developed from multicarpellary gynosium or where carpels are fused together. In simple way, when carpels are fused together is developed in aggregative fruits. For example, raspberry is the aggregative fruits. The next type fruit is the multiple fruits, which develops from the flower of whole inflorescence. This type fruits called multiple fruits. Mulberry is the example of multiple fruits. In parthenocarpy fruits is the most important process of in this time development of fruits. Several edible fruits such as seedless, grapes, orange develop to afford of the taste because these are fruitless, these are seedless fruits. but these did not use to raise offsprings. So these are obtained only of a taste but not in other generation development. In parthenocarpic fruits does not have an embryo and endosperm because these fruits are seedless. So endosperm and embryo is not developed in parthenocarpus fruits. One important process of which helps the parthenocarpus process is called apomixis. Apomixis is the phenomenon which proceeds seeds without fertilization. This phenomenon does not include formation or mixing of gametes. So, it does not produce genetic variations. This process is called apomixis and it helps the preserve the useful of parental characters. So here we stopped the development of embryo and types of fruits after embryo development. And write down the today's homework. Question first, what is the cotyledons and what is important of the cotyledons of seeds? Next question is, draw a diagram of a mature dicot embryo. The next is, why an apple called a false fruits but banana is called parthenocarpic fruits? The next question is, what is endosperm and explain its significations. And next question is, what is apomixis and what is difference between apomixis and parthenocarpus process? This chapter was finished and next topic is the human reproduction in the next video. Thank you. Have a nice day.